This episode of the Business of Biotech is brought to you by Avantor. Every day, science has the power to change lives for the better. Avantor partners with the scientific community, pioneers and innovators in the lab and in bioprocessing to help solve complex scientific and operational challenges. Avantor delivers end-to-end support from discovery to commercialization for the global biotech and biopharma ecosystem, providing mission-critical products and services that are integral to advancing life-changing innovations for patients. Visit avantorsciences.com for more information. Welcome back to the Business of Biotech. I'm your host, Ben Comer, Chief Editor at Life Science Leader, and I'm thrilled to speak today with Tom Levenson, a professor at MIT's graduate program in science writing and the author of 7 books on diverse topics, including The Science of Climate, A Musical History of Science, Isaac Newton's investigative work for the Royal Mint in London, Albert Einstein's Berlin period, the search for the hypothetical planet Vulcan, and other topics. He's also the recipient of a Guggenheim Fellowship and has a Peabody Award to his credit for work he did on a series for PBS. Tom just recently published at the end of April a book on the history of germ theory called So Very Small: How Humans Discovered the Microcosmos, Defeated Germs, and May Still Lose the War Against Infectious Diseases. We're going to dive into some of the themes, people, and issues Tom writes about in So Very Small, because the history of germ theory has important implications for public health today, as well as the development of new antibiotics and therapeutics. What's past is prologue, as Shakespeare wrote, and with the shakeup happening currently at NIH, HHS, and FDA, I think it's a timely conversation. Thanks so much for being here, Tom.
Thank you for having me. It's great to have a chance to talk with you.
I thought we could start with the genesis of So Very Small. What made you want to write it?
Well, I kind of have to blame my wonderful London-based editor, Neil Belton, who is a formidable nonfiction editor and currently head of an imprint for Bloomsbury called Head of Zeus. And in 2012, I published one book with him, and I was over in London for a variety of reasons. We met up for a coffee. And he told me that he was working on this series of books that were supposed to be very short, 25,000 words or something, on decisive moments, some like single incident that then could be, used to look at something larger. And so, for example, he had the first production of Handel's Messiah, which opens up all kinds of interesting doors into, you know, music and, the sort of social structure, all that kind of stuff. And another example he gave me was a look at the creation of Picasso's painting Guernica, about the Nazi bombing of this Spanish town during the Spanish Civil War. That was kind of a trial run for later blitzkrieg tactics. And these were great books. And he had a bunch of others that he had on his list. And he's a very innovative editor. And he said, we don't have any science. Got to give me some science. And so at that time, I had just finished the Newton book you had mentioned, and I was looking at some sort of some new stuff that also had some kinds of physics connections. And I said, well, there's an obvious one. When Albert Einstein goes up the mountain at Mount Wilson in 1930, and he'd come back, where the universe has changed. He's seen the first images that say the universe is expanding. And that's a complete, that's a real radical change in our understanding of, reality on the larger scale. I said, the other thing that and I said, I'm not the person to write this. I'm not a biology writer. I don't, I'm not studying this, but it's really been fascinating to me all these years that you have the germ theory revolution in the late 19th century. Depending on, you can sort of put the dates where you want, but by convention, the date that Robert Koch, one of the two sort of giant figures that are associated with the germ theory moment in particular, the other being Louis Pasteur. Koch figures out the life cycle of the anthrax bacillum and demonstrates decisively that bacillum, B. anthracis, is in fact the pathogen that brings about a case of anthrax. And that happens, starting 3 days before Christmas in 1876, and he sort of finishes the experiments before the end of the next January. And so like that 1870s, 1880s, 1890s decisive moment before disease and our understanding of our relationship to the worlds of world's biology, the whole idea of the causation of disease, all these kinds of things is one way, and afterwards it's different, and it's different in ways that give human beings huge advantages in the millennia long struggle with infectious disease. So this is a great topic. Somebody should write this. Go find somebody to write it. And he never did. The series came and went, all that was done. And he and I would meet over the years, and I did 2 other books with him and with my American editors. And we would occasionally talk about it. And I have to say, I didn't know that there was really a full book there until I started thinking, well, how do you get to the moment where Koch, figures out anthrax? Just taking that as that sort of marker.
And that was a story that you were already familiar with or already knew, the Koch and anthrax discovery?
I knew I'm a magpie of a science writer, and I read very widely, and so I know a little bit about a lot of the history of science. But I wouldn't have called myself deeply knowledgeable. I feel like I know it pretty well now, but I didn't. I was not more than kind of 1870s newspaper reader level of knowledge up to that point. But what I noticed is the critical thing was obviously the identification of microbes, and specific as the agents of disease, and specifically that each disease was associated with a single microbe. So there was a very carefully constructed chain of cause and effect. And that's the great breakthrough that leads to all kinds of possibilities in responding to these infectious diseases, all kinds of great things that we'll talk about, I'm sure, in a few minutes. But it turns, of course, on identifying the fact that microbes exist at all, right? And I was thinking, all right, well, when were bacteria discovered? And this I actually knew pretty well, because I've spent a lot of time in the 17th century and spent a lot of time in the early history of the Royal Society. And basically, microbes were first seen in 1676 by this Dutch draper, cloth merchant named Antony van Leeuwenhoek. Again, apologies to anyone who speaks Dutch out there for my butchering of that name, I'm sure. But sort of like 200 years, almost to the day, runs between the first observation of microbes, bacteria, and the demonstration that they actually matter to us in a really consequential way. And I said, that's a long time. Why did it take so long? And that was the question that got the book started. Why did it take so long? It made it a really interesting and bigger project, I think.
Yeah, how do you approach the research? I guess using this book as an example, when you look at that 200-year-long period where as you said, the 1660s, I'm going to butcher this as well, but Van Leeuwenhoek is sort of looking at these, little squigglies as a curioso, as almost as a form of parlor entertainment, and then 200 years go by before that connection is drawn between these microbes and infectious disease. How do you dive into that? How do you get started?
Ooh, well, that's I sort of feel like as my professor hat goes on, and it's like, don't try this at home, kids, because I'm somebody who spends a lot of time. I like primary sources. I like reading the early philosophical transactions of the Royal Society and Leeuwenhoek's letters and so forth. All of Leeuwenhoek's, not all of them, but an enormous corpus of Leeuwenhoek's letters to the Royal Society and to others are online. You can just, in English translation, parallel Dutch and English, and you can just go read them, play around with them, see what he was looking at, see what he was thinking about, see how he expressed himself. There were, there's a body of history of science work on germ theory, of course, and on the discovery of microbes, history of biology. And so I'll both look at some of the, just to get started, I'll start, getting some of people I know will be characters in the book, and I'll try and get their words and their thoughts and their stories as just part of in my head and part of the sort of thinking through how the storytelling in the book tries to make some arguments, and I hope some fairly compelling and useful arguments, but it does so by telling stories that you can then draw meaning out of. It's not just sort of you know an academic essay. I don't work that way. So getting in touch with the people who are involved in the work is really an important first step. And the other first step is to really try and see what the arc of the book is, where, how this in particular, um, this 200 years of halting discovery went from just seeing microbes to recognizing one of the important things that they mean for us, mean for the earth. So I spend some time with some of the scholarly literature on it. And then the thing I do, and I do this quite early on, I do it as I'm writing the proposal for the book, which is, about 10,000, between 6 and 10,000 words usually. So it's a fairly substantial document in itself. I really try and think through what the arc of the book is, as a matter of plot. What incidents are important to the development of this idea? And once I've got those, then it becomes quite simple. Or simple as in the task is simple. The work may not be easy, but if one thing I want to do is find out how the 17th century reacted to the discovery of microbes, and it is, as you say, primarily as kind of a drawing room curiosity, much more than this sort of enormous scientific breakthrough. Which I have to say, I was talking with somebody about this recently. And it's still kind of bizarre. I mean, think what our reaction today would be to have real proof of life on other planets.
Right.
It'd be a huge deal. In the 17th century, no one had any idea that there was this whole realm of living creatures, beyond our senses, inaccessible to us with the naked eye. And yet there they were. They were discovered. They could be seen by others once Leeuwenhoek showed them how. An entire previously unexpected domain of life. How was that not a big deal? I still don't, I don't quite, the past is a different country. They do things differently there, but that still is kind of amazing to me.
It's that's one of the things that I found really compelling about the book, were the way that you stitched these stories of individual people making discoveries, and then trying to take them forward, sometimes against pretty remarkable reactions or pushback from society in general, and even from other scientists. I mean, one of the themes that I wanted to ask you about in this book, I think, touches on the hubris of human beings. I mean, the cultural influence of Christianity runs very deep in Western culture. In the Bible, God makes humans, in his image, grants them dominion over every living thing on earth, which gives rise to the great chain of being concept, which you discuss, essentially a hierarchy developed in the Middle Ages, placing humans below God and angels, but above animals, plants, and minerals. And it's the idea that we're not of nature, but above and apart from it. And so I wanted to ask, if what you might say about that, how human hubris has played a role in the history of germ theory acceptance and its practice.
I think, as you just summarized, one of the big points in the book. It's very hard to imagine that these lovely, cute little squiggling things that Leeuwenhoek saw could matter to us if your whole cultural framework says, agency flows down. God has ultimate agency, and he grants it to it, whatever, the gender of God, passes it on to us, and we, as God's agents. And in the 17th century, you had people, who really understood themselves to be, in some senses, God's agents, or even, mortal angels on earth. There's some beautiful writing about, how this Isaac Newton himself had this sense of his role as directly deputized by God in some ways. Very deeply held. I would say it, I think in Western in European and North American culture, the expansion of the European world. Yes, the Christian sort of framework has enormous and lasting cultural significance. But the idea of human exceptionalism is one that's expressed in other cultures as well. Um, but it's that sense that we are uniquely empowered in this world. We have big brains and opposing thumbs, and we're a tool using. And God has given us this beautiful created world in which we can do things like make our living through agriculture or hunting or whatever it may be. And I think that made it extremely difficult to recognize that something is diminutive and seemingly unimportant. I mean, they had not knowing about them, hadn't seemed to make a difference to us up to that point. I think it's very hard to imagine those little critters, swimming in a drop of water as having agency on their own, having some way to get at us. And I think there are two things. First of all, I think that human exceptionalism, even when the explicitly Christian framing of it drops away, that lasts into our own day. I mean, that's still a very powerful, I think, cultural impulse. And you can see it across, all kinds of things, including our politics right now. You mentioned changes at NIH and so forth. I think some of the sort of deep, where the license to do that comes from for the people who are trying to make these changes comes from, I think, that sense of human beings as the actors and everything else is the acted upon. So that continues, and I also think that the great chain of being in that sort of hierarchy of creation is something that people applied as a notion to all kinds of other things, including human society. So in European world culture, upper class white men are at the top, and everything else flows downhill, and that becomes a problem when you look at some very specific incidents in the history of germ theory, as for example, when not just the famous story in Vienna, but the with Ignaz Semmelweis, but the earlier folks who figured out that puerperal fever, childbed fever, this infection that kills women who have just given birth, there was a pretty clear series of demonstrations that this was a problem caused in some way by the doctors or midwives, the medical people attending those births. And people were able to document, as early as the late 18th century, that this was most likely something that was transmitted. They didn't know what it was being transmitted and so forth, but the sort of epidemial, the disease detective links between a doctor or midwife and a chain of patients who would all suffer these infections was really clear. It was established well. And in response to one of these, Oliver Wendell Holmes Sr., not the Supreme Court justice who was his son, but the dad who was a poet and doctor and other things, early in his career in the 1840s, said this. And the response, he was a young doctor at the time, and he was not a specialist in maternal care. And the sort of leading textbook writing OBGYN doctor of the day in Philadelphia responded, no. That's the maunderings of a sophomore. Couldn't be. And because this is a quote, a gentleman's hands are clean. So the idea of a hierarchy is there. Yeah.
Yeah, just by dint of being a gentleman. Yeah, and I wanted to touch on some of the other historical misconceptions that have functioned as a block or a resistance to acceptance of the scientific research. I wonder if you could explain maybe briefly miasmas and what role that concept played.
Sure. I mean, think about, an infectious disease outbreak before, like, for example, the incident that starts the book, the Great Plague of London. So plague shows up in 1665. There's there's an index case that probably isn't actually an index case, but there's an early case that people refer to in a poor parish. And then over the next couple of months, a few more cases pop up, and then the disease becomes much more widespread, and the death tolls start rising until you get in what was still a quite small, I mean, a fairly small city by modern standards, enormous death tolls, 1,000, 2,000 people a week, whatever. Um, and you needed to explain two things. How an individual case of the plague got started, and then how did it spread? Um, and there are a number of there were a number of ideas about mechanisms of disease. That some ideas that would, from classical antiquity about, keeping the body in balance, the four humors. Some more seemingly modern, though still with much of the same apparatus, so-called chemical medicine, where it was a more specific, some more specific substance that was in or out of balance. So that was how the disease, that was what the definition of disease was: something that caused your body to start running awry, which is, and this is important to understanding why this idea was able to last so long. At least as a metaphor, that's not a bad, way to think about a
Right.
disease, right? Think about diabetes. Your insulin metabolism doesn't work anymore. Your insulin levels go out of balance, and you're in trouble. And you treat it by, you know, with by one way or another, bringing those insulin levels back into a safe range. You know, Everything we know about the mechanism of diabetes is different from what people know about the plague in 1665. But that sort of metaphorical level, you can sort of see how it's an effective way to frame a disease,
An
persuasive.
imbalance, right.
Yeah. And similarly, for the contagion, for how to get it from place to place, how does it get to a person in the first place, and then how does it travel? A miasma literally means bad air. Um, and so the idea was that there is some, if you believe it, it's that every disease is simply God's judgment. God sort of opens the fissures of the earth, and, foul air bearing disease, balance, unbalancing stuff goes out. And, it allows for a disease to arrive someplace. And, if it's really in that sort of sense airborne, you can see how it would spread. They did in that period use the term and have the concept of contagion, which the root means literally to touch. So you would touch something that was uh, affected by a disease, and that touch would communicate something that could then be passed on. But by the 17th century, certainly contagion had been sort of loosened as a concept, had been loosened and sort of broadened to the idea that proximity rather than actual necessarily sort of mechanical touch was necessary. But once you have miasmas, you have a way for the disease, that contagion, to occur and keep passing from person to person. And the important thing to understand is that you look at a disease spreading through a city, as the plague did through London in that period. And you combine the models of disease itself as an unbalancing, and that the seeds of disease, whatever they may be, they use terms like that, seeds of disease, could be carried sort of invisibly on these gusts of air, or air blowing it onto the person of somebody who's in a sick room, and carries it with them, and a breeze blows it to the next person. However you want to start mapping out the individual scenarios in your head, it's not a bad description. And so the ideas persist in part because, even if they don't tell you really what to do about an illness, other than sort of attempt to bring things into balance by, cooling people or heating people or giving people beer or, bloodletting or whatever it may be, these some way to bring one of the bodily systems into balance. There's no specific mechanism, so there's no specific treatment that's available. But if you're just trying to paint a picture of what seems to be happening, that does a pretty good job. And that's sort of one of the big problems for getting to germ theory, is you have to come up with something that's not just more correct, but is in some ways more persuasive, more directly supported, has some leads to some direct possibility of action. And that takes a long time, especially as, in some senses, miasma theory is, quote, true. I mean, yeah, if you live on top of a sewer, if you don't have, proper hygiene and all these kinds of things, the bad smells and the, decaying flesh and all this sort of stuff are signals that something's going on. Miasma theory doesn't tell you what is going on, but it's close enough to be difficult to dislodge.
Right, right. And it creates essentially a barrier for someone who is making a discovery and attempting to go against that now commonly accepted wisdom. I wonder, and there were a number of names that I had not heard in this book, which is another reason I really liked it. I wonder if you could give an example or two of some of the maybe lesser sung historical figures who helped develop the science behind germ theory. And you talk about a number of different diseases in the book, so I'll let you choose.
Well, I think consistently the figure that people are most surprised about hearing is Cotton Mather.
Yeah.
We know Cotton Mather as this, I live here in the Boston area, and Cotton Mather is our, hellfire and brimstone, witch burning, Puritan Bible thumper. And, he was all those things, but he was also somebody who apparently as a young man actually thought about becoming a doctor, was profoundly interested in he didn't have a disciplined mind. He was a complete magpie, but he was profoundly interested in the natural world. He became a fellow of the Royal Society. He sent them, lots of letters about sort of American curiosities, some of which were, clearly sort of, he was credulous about wild reports, but some of it was very, a lot of natural history. He was fascinated with medicine. He composed an entire book called The Angel of Bethesda. That was manuscript. He left it as manuscript when he died, and it wasn't actually discovered and published until the 20th century. But it's a sort of an encyclopedia of everything that he could gather about healthcare and medicine, how you would prepare recipes for this or that, and it contains some really interesting things about germ theory. In fact, actually, to me, very important. He comes into the book because he is one of the two great sort of English-speaking pioneers of immunization. It's not true vaccination, but it's the predecessor. It's the precursor to modern vaccination, inoculation with smallpox. He learned about that process from his enslaved servant, a man he named Onesimus, who had been born in Africa and taken from Africa to Boston, and then gifted to Mather by some of his parishioners. And Onesimus had smallpox inoculation. He received that in Africa. And so when Mather asked him if he had smallpox, Mather records his answer as yes and no. Didn't have it in the wild, had a reaction to the smallpox inoculation, and is now immune. Shortly after, a few years after that conversation, an Italian doctor working in Constantinople, in the Ottoman Empire, reports that the Turkish medicine does the same thing, and that was published in the Royal Society's Transactions. And Mather saw it and wrote to the secretary of the Royal Society saying, "I can, affirm this. I've heard this story from my slave and from other people who've had contact with Africa. And the next time smallpox comes to Boston, I want to try it here," which he did to significant success and significant opposition. I mean, anti-vax sentiment starts right at the beginning. It's still, it's still with us and has its roots in the reactions to these experiments in the early 18th century. And so that's one of the ways Mather gets into the story. The other way is this remarkable, in this Angel from Bethesda manuscript, he writes down explicitly the speculation that these microbes that Leeuwenhoek, and he names Leeuwenhoek, by name, cites him. These microbes, the term was animalcules, these little animals, as they were known, that Leeuwenhoek has identified, might, they're everywhere. They're on leaves, or this and the other thing, and they could be the sources of specific diseases, and we should really look at that. And to me, that's both fascinating. This is in like 1720, 1721, something like this. So
Amazing.
150 years before germ theory proper actually makes its way, into science, as it were. Um, and it's important to me because it shows that the difficulty in working out germ theory wasn't simply, and perhaps for much of this period, not mostly scientific. It was one of finding the ways to ask the right questions and then pursue it. Mather was not an experimentalist. He was not a disciplined man, disciplined scientific mind. Um, so he wasn't the person to follow that up, but he could think it, right? And there was at least one or two other people who were thinking the same thoughts then, but they were stillborn thoughts. Nothing happened, and that to me is one of the things I really want to understand in the course of the story I tell in the book.
I think it's easy in 2025 to looking back at history and seeing, the gentleman's hand is always clean. Miasma we just talked about. Looking backward and saying, wow, it's incredible that, these brilliant discoverers and people who made these forward leaps in germ theory faced resistance. We couldn't be in a situation like that today. We couldn't have those same kinds of dogmas and biases in 2025. You write in the book, it's hard to recognize what one's assumptions omit, all the more so when those assumptions are born of individuals and a society's shared beliefs about the interplay of the natural and human worlds. My question is, you know, they're difficult to identify. I think that should go without saying, but are there some existing biases or dogmas today in medical or scientific research, and how might we go about better detecting them?
That's a huge question, and it's tricky because some of the issues are with scientific research, and some of them are with, sort of the questions for the larger, you only do the science that a society is willing to entertain, right? And we're seeing
Right?
that explicitly. Um, there was an election in November. The results of that election were what they were, and there are decisions being made about what the NIH should be doing, what the NSF should be doing, and so on. Basically, every federal government science research agency, um, and you can only do and that's in some sense that's the expression of what a substantial chunk, enough of a chunk of our society now to achieve political power. That's what they think is important or unimportant. And that's not necessarily what a scientist, an individual scientist, or a discipline as a consensus or near consensus might find important. Um, so right now, for example, you have explicitly a shifting of NIH attention, Health and Human Services attention, from infectious diseases to so-called chronic diseases. That's a choice. I think it's a wrong choice. I think it's a terribly dangerous and scary choice. But that tells you that there's an assumption there. There are that, and again, like most assumptions throughout the history of all this, it's not altogether wrong. That there are the 21st century intensely urbanized, very sort of quote mediated in all kinds of ways, not just media, but our food is mediated through a whole bunch of systems and all this kind of thing.
Right, a more sedentary workforce, yeah.
All that sort of stuff. So it's not wrong to say that this has some consequences and it should be, it's important to study. But um, B. anthracis is still out there. Yersinia pestis, the plague microbe, is still out there. V. cholerae, cholera microbe, is still out there. The smallpox virus is not. That's extinct in the wild. But polio is still just out there and could, under certain circumstances, roar back. Tuberculosis is very much there, killing 1 million people a year. So if you say that chronic diseases are worth focusing on, that's an unexceptional comment. If you say chronic diseases are the essential problem, microbes get a vote, right?
Yeah.
You know,
Or
we
don't,
can,
right.
yeah, we may wish that infectious disease um, was not a potential threat, but you know, that and a token gets you on the subway, as it were. Um, I think one important thing to remember is we may know more facts. We may have more um, coherent and in some ways rigorous theories that we use to organize those facts than people did in the past. It's not we may, we do. That's we understand infectious disease down to the level of molecules. And individual changes in genomes that produce different pathogenic effects, all these kinds of things, and our predecessors did not. But that knowledge isn't the same as saying that we are smarter than our predecessors, or that our habits of mind are somehow cleansed and more rigorous. They're not. We're not smarter than Isaac Newton and Robert Boyle and all those folks, Huygens or whoever you want to throw
Yeah,
in
we have
there.
the same hardware.
Yeah, and so it would be presumptuous and of me as a popular science historian to say these are the assumptions that people are making in science and medicine that are clearly going to bite us in the butt. Um, but I feel pretty confident saying that those tricky assumptions exist.
Are there parallels that you see between mapping the human genome and mapping the microcosmos?
Um, yes, and I see them both as it's doing so produces this extraordinary trove of facts, and I think one of the things that's most fascinating to me about the Human Genome Project, I'm not in my first youth, as you can tell from my hair color and all that. Um, I'm old enough to remember, and I started work as a science writer in the 1980s. And it was a big deal to sequence a single gene and to identify its protein. You'd look for the gene for sickle cell trait or the gene for Huntington's or whatever, and very intense search for these particular sequences of DNA. And a whole PhD thesis would be a graduate student working through one gene or even part of one gene because it was so laborious and so slow. And in that context, people said, "Oh, great, we sequence we build the technology, we do all this sort of stuff, and we sequence the whole human genome, and then we'll know how to build a human being, and we'll understand all these diseases, and we'll be able to do all this stuff." And so they sequenced the whole human genome, and with each successive level of detail and precision in our understanding of the basic, information environment of molecular biology, how DNA works in us or in, a planarium, the C. elegans flatworm or, whoever, chimpanzee. Um, what's been striking is how much more complex it turns out to be. Our assumptions about what kinds of knowledge and power we will get out of doing, say, sequencing the human genome, has we now know. I mean, I was just seeing a marvelous paper that suggested that there is an important, RNA mechanism for deciding which of a couple of different, or which of a number of different proteins sequences of DNA may produce. That it sort of dictates which bits of DNA get expressed, and you make different proteins. And this is one of the ways you get to build something as complex as you or me, despite the fact that we only have 20 or 30,000 genes, which was one of the first shocking and very difficult to understand results of doing the Human Genome Project. How could you make us out of so few genes, especially when seemingly simpler creatures in some cases had many more genes? What's that all about? And I think that the same is true of the microbial world. Cataloging the entire microbial world, especially if you include, as modern usage does, viruses as a subset of the microbial world. You do all that. I mean, there's a question of what it means to catalog them, identifying strains, doing genomes for each of them. There are lots of things you can do. Enormous amounts of useful stuff to come from that. At the same time, I think it will expose us to enormously fascinating problems that simply doing that cataloging will not be sufficient to help us figure out.
And I think a similar thing is happening with to get back to microbes, the microbiome. There were a number of drug companies who, in the last 10 years or maybe 10 years ago, there was a real effort to design drugs that targeted the microbiome for various conditions. And I think people found out, because of the clinical failures, that system is much more complicated than we initially understood, and it's still, I think, largely a mystery. But
Yeah.
is that germ theory's next frontier, the microbiome?
I I think I think certainly it's of interest. I mean, one of the things that's fascinating is, all right, you have something like, name your poison, the rabies virus, or V. cholerae, cholera, whatever. And there's a sort of relatively simple sequence of cause and effect. You know, you get infected with this particular microbe. Certain, recognizable things happen. We now, as we didn't then, but we now know, for example, what it is, what specific molecular events the cholera microbe sets in motion to start, sloughing the lining of your intestines and your guts and all that, dehydrating you and all these terrible things. And we understand that at a mechanistic level pretty well, very well. So that's one thing. We have these like sort of point source individual microbes doing individual things in your body. But it was, as you're saying, with this rise of sort of microbiomic medicine, the realization that we have, more bacterial cells, microbial cells in our body than we have human cells, that we live, in some ways we're just the tube that we're the chauffeur for all these microbes. We just carry them around the world, and they're doing their thing and using us for their ends. But they're also symbiotes, obviously, the gut microbiome is crucial to your digestion and your digestive health. If that goes awry, you get all kinds of bad things. And when that's healthy, you don't have ulcers. You have, all these, desired outcomes occur. And this recognition that there is this intensely complex ecosystem, this whole, again, sort of microbial microcosmos within our own bodies, within each of our own bodies, that when it's healthy is serving essential functions for us. And you can obviously see examples throughout the kingdom of life. Microbes that do great things to allow plants to fix nitrogen. Microbes, there are all kinds of different ways in which sort of macro scale organisms interact with these micro scale ones in ways that are productive for both. And in some senses, the diseases that result from some disruption of one's microbiome are conditions that may, sort of require a kind of germ theory like breakthrough to understand. Is it the extension of germ theory specifically? Well, germ theory was really built around isolating individual microbes and going through a series of steps to rigorously show what their properties were and what their effects were on whatever they have to infect. And I don't think that simple, sort of fairly straightforward experimental protocol is going to really be the driver of what is, I think, a much more complex, much more multidimensional systems biology problem.
Right, they're not working in isolation, and there's been some really fascinating research done on the gut-brain axis with implications for diseases like Parkinson's and Alzheimer's disease. So yeah, I mean it seems like a really difficult one to even study. I wanted to ask you about antibiotics. You raised the issue of responsible use of antibiotics in so very small, as well as the implications of antibiotic resistance. What kind of policies or funding mechanisms do you think are needed to adequately address this issue?
Okay. Well, it's first of all, I'd like to say, shout out to some of the people I've talked to over the years, or stuff I've read, who've helped me understand it. Microbial resistance. Maren McKenna's writing has been really great, and she's an incredibly powerful thinker and reporter on this stuff. And there are lots of others. Laurie Garrett has written very well about this stuff. And so nothing I say is unique or original to me. These are thoughts that people have been working out for a long time. That said, the antibiotics as a drug that a for-profit drug discovery system is going to produce, that's a real problem. And it's a problem for a couple of reasons. One is antibiotic resistance means that any new antibiotic has either a finite lifespan or real constraints on how you use it in order to preserve its effectiveness when resistance is so readily developed and then it spread. And the other thing is that antibiotics, even antibiotics that are effective, the great thing about them is they work.
Right.
You get an ear infection, or your kid gets an ear infection, you give them a week or two of some drops or a pill, and they're better.
They're
And
not
that's all
administered chronically. Exactly.
right. They're not statins, right?
Right.
And so this is not a criticism of a for-profit drug company. It's just a fact. These are not the kinds of compounds that those kinds of institutions are set up to develop. So yes, that means, yet that said, the economic return on on a successful antibiotic is enormous.
Right.
All those lives saved, all those sick days, eliminated, all that sort of stuff. The benefit to society is huge and can be counted in money terms, and it's huge in money terms. So these are drugs that have a clear dollars and cents, as well as it's good not for human beings not to die. It's good for human beings not to suffer. There's a moral and just kind of like just sort of a civic value to it. But there's also if we are going to be with, as the old phrase has it, homo economicus, if we're going to think that way, it makes sense on that sort of accounting level as well. And if individual, if Merck or GlaxoSmithKline Beecham or whoever it may be isn't set up to do that, then somebody else has to, and that somebody else has to be essentially a social commitment. We have to decide we're going to spend our money, our tax dollars on it. We're going to develop research. We're going to support nonprofit. It could be governmental, could be non-governmental, doesn't matter. But we need to have an antibiotic and more broadly antimicrobial drug research program that is something we, as an individual nation, the United States, as a developed world with the resources to do this, that's something we need to pay for. And we need to pay for it not just because it's the right thing to do, it not just because it will save lots of lives, but because it's part of creating a sustainable, flourishing, thriving, and economically, as well as by any other measure, successful society.
And ideally, a majority, and I'm just speaking about the U.S. here, but ideally, a majority of people would come together and demand that tax dollars be spent to research and develop these new antibiotics. That so far has been difficult to generate. I mean, maybe another option is angel investors, the exceedingly rich, are being talked into, maybe instead of investing in space travel and investing in the development of new antibiotics. Which one is more likely to happen is what I guess what I'm getting at. Is it convincing enough people, or is it convincing the rich?
I mean, I'm certainly not against trying to persuade Bill Gates, who already is very active philanthropically in the healthcare space, but Jeff Bezos and whoever, to put as much money as they care to, as much money as they can, into antibiotic research and basic science, say basic biological or sciences, whatever, because many of these things derive, the translational gap between curiosity-driven basic discovery and developments that directly benefit human beings, that gap has been shrinking. And many of the great ideas that then serve human beings well come from rapidly emerge from stuff that you can't predict, but are can't predict individually, but that we know from decades now of experience. This sort of curiosity-driven basic science is the seed corn for all kinds of technology development that sort of flows downhill from it. So yeah, absolutely. Get those billionaires to give back in that way. But I don't think that's a reliable, sustainable source of what we really need. Science is not, part of the problem with billionaires or anyone is, you saw it, for example, in Silicon Valley, people investing in sort of media ventures. They'd be really interested in, right? And then after a while, you say, okay, well, I did that. It's not sustaining on its own or whatever. I'm going away. And the enterprise goes away, and all of a sudden you have a gap where that used to be. If you do that in science, the problem is not just that an experiment that's happening at the moment the funding goes away stops. I'm sure and your audience knows much better than I, but really good labs have developed over time institutional culture, set of sort of informal knowledge, something as maybe as seemingly silly as that cell sorter over there needs to be encouraged in this way to do its job right, and just how the machines work or whatever. But there's also sort of a collective act of thinking that goes on in productive, interesting labs. And you pull the funding and those people scatter and the machines go away and the space gets reused. It takes just to set up a good lab to do really cutting edge work in a lot of these now increasingly precise and sort of nanoscale or microscale disciplines. Just the physical act of setting up the lab is could be a year or even more in some cases. And the informal knowledge is more than that. It takes a while to build back a team that really knows, can sort of communicate in efficient and productive ways. So relying on billionaires who may come and go, whose interest may wane when a result doesn't happen and so forth, unless those billionaires are willing to endow institutions, sort of what happened with the Broad Institute and some other the Weiss Institute at Harvard, places like that. They actually have endowments and they can sustain themselves. People are willing to do that. That's great. But truly, the scale of science these days and the scale of our societies, where US is 330 some million people, many $1 trillion a year economy. If you want to make a difference on that scale, you need sort of social level funding. And I think you need to do so in a way that is not subject to the each change of administration's whims. I mean, the idea not just that you're changing the focus of NIH or NSF, but that you're canceling 400 grants that have already been awarded on which work is being done. Those labs will shut down. They'll lose all that institutional infrastructure, that capacity. And the hit to U.S. science and the hit to our ability to develop new antibiotics, to develop new vaccines, to look for new ways of addressing microbial issues altogether, that will be set back for years to decades. And that will have not just American impact, it will hurt us, hurt our economy and so forth, but obviously it has worldwide impact and it has life and death impact on a lot of people.
Right. And a number of the endowments coming in from family offices and people are often targeted to a specific disease. You know, they're focused on cancer or cardiology or a rare disease. Not so much, you know, large endowments for basic science. I mean, the NIH, I think, does the vast majority of that kind of work or has done that kind of work in the past. Are there any other, I guess, potential consequences that you could name around funding cuts to the NIH? What's happening there right now?
Well, I think again, I don't know more than what any of your audience knows from careful reading of the newspapers, and then perhaps the next two or three levels down of more specialized publications. Clearly, shutting off funding for some scientific research or a lot of scientific research directly affects those particular research programs. Cutting off the science educational pipeline, which is one of the things that happens when you cut funding for scientific research. You don't get graduate students in, you don't have as many graduate student places. Undergraduates, universities lose the ability to educate undergraduates, certainly undergraduates who need scholarships and all that kind of stuff. So you're going to have a sort of broader based skill loss that'll happen. That's likely to happen. Another thing that's going on is a lot of the just reading this morning before we got on this conversation about a bunch of the sort of data collection that the federal government is doing across a wide range of domains, from food poisoning to weather disasters and everything in between, right?
Yeah, disease
A lot of that
monitoring.
data, a lot of that going away, at least temporarily. And that means two things. One is bad things can happen out there and we won't know about it and therefore can't address it or learn from it. I mean, there's one of the things about disasters is they teach you something, at great cost. But we won't even be able to do that because we won't know, the scientific community, the research community won't know that, an event has happened because this monitoring that we've come to rely on has gone away. And I don't see how you easily replace that. I mean, we just have to, I think, fight those decisions. They're ill-judged, I think. It's one thing to say, I don't want to do, I don't want to research cancer. I want to research obesity. Look, then Vice President Biden wanted a moonshot on cancer. That was a decision about what direction to go. And there were, I think, a lot of scientists who said, as with the war on cancer, the original Nixon war on cancer, that it's a mistake to just put all your focus on that singular complex of diseases. There's lots of other things that we need to know, including the basic science that will help illuminate cancer and lots of other stuff. So there are choices that different administration makes, and okay, they get elected, they should make those decisions, but that sort of unilaterally disarming our ability just to find out stuff, either about nature or about what's happening in our society as nature has its way with us, that leaves us in an extremely vulnerable position. And I'm not sure I've answered your question, but I will say that makes me quite scared.
For our audience, listeners in leadership positions at biotech companies, at pharma companies, are there any lessons, any cautionary tales, any messages that you would hope they might glean from So Very Small?
Well, I think one is that you should probably it's really hard for a startup, I know, which is always just trying to get everything done before the money runs out. But there's this, you got to have a little room in your hip pocket for something unexpected. I think some of the critical, there were these interesting ways by which people made the inferences that finally led to germ theory. Some was, just repeatedly identifying the presence of a microbe in a disease without knowing what the mechanism was. And another was, really doing a complete reconstruction of, for example, how cholera propagates through a population that was essentially all of germ theory except for the germ itself. these were people were able to work with these ideas and think them through, and they provided a foundation for real breakthroughs to come. So I think, that saving a little bit of budget, a little bit of time, allowing a certain number of projects that may be not quite directly on the main line of your applied research programs is probably a useful thing. More broadly, I would say that I think the uh, the phenomenon of infectious disease is something we have largely allowed ourselves to forget about because of the extraordinary success of vaccines against all the common diseases of childhood and many other diseases, and the power of antibiotics since the end of World War II.
It's a
So,
really good point, and I think I'll just quickly interject here. The primary source accounts of people dying from cholera in the book were so shocking to me because, it's something that we haven't seen here in my lifetime in the US. And just the speed at which they died, people from all walks of life, it was really shocking in a way to read about that, how people experienced it in real time.
absolutely, and I mean, I think the COVID pandemic was kind of a warning because terrible as it was, as disruptive as it was, as many people around the world who died of it, it was, as a novel disease goes, it had what a roughly, before the advent of the vaccine and so forth, it had a roughly 1%, I think, mortality rate, something like that. I mean, uh, forgive me if my memory is playing tricks, but I think it was in that range, which is terrible, but not society devastating, right?
Right.
You know, imagine, and oh, by the way, to me, COVID is one of these great, both tragic stories of failure because of the, ultimately political resistance to basic public health that led to many more unnecessary cases and deaths, and this extraordinary triumph of science. I mean, to go from the public announcement of a disease in December to its entire genome being sequenced by within January, so within weeks, not months, to um, a basic vaccine design that's there and manufacturable by, I think, April, and is in people's arms a couple months after that as a test, to widespread manufacture and distribution by the end of the year, so less than a year after the first emergence of the disease. I mean, that is a testimony to the extraordinary capacity of modern molecular biology, virology, um, and to the industrial and intellectual capacity of both the government and the various startup and established pharma firms that were involved in getting that vaccine out. I mean, that was a triumph. Um, what I worry about is the next emergent disease may have similar transmissibility to COVID, may have the same global reach because air travel is, part of our fabric now, but has a mortality rate or, has, more severe consequences, though, long COVID and so forth is pretty darn severe, but and a higher mortality rate. Imagine if COVID had, say, a 3% mortality rate instead of 1. So you catch the disease and you have a 1 in 33 chance of dying. That would be, I think, just that change, doesn't sound huge, would be enough to be, I think, a much more socially disruptive. I mean, obviously tragic for the individuals and families involved, but imagine if you look around and 1 in 3 is gone. The so the in some sense, I think and COVID was, as I say, this sort of relatively mild warning of, reminder that the microbial world, the viral world, is still out there and still interacts with us in ways that we don't always love. And I think one of the things that would be really worth your listenership's attention is to think hard about the kinds of infectious disease threats that people who look at things like zoonotic diseases, diseases that leap from animals to people. Um, and perhaps some of the more conventional or more familiar older diseases like measles, which is making a comeback because our vaccination levels are dropping. All that, those are areas of both fascinating scientific inquiry and possibly important commercial and work as well.
Absolutely. Well, we are running short on time here. Tom, it's been a real pleasure speaking with you. I really appreciate you being here. Tom Levenson is a professor at MIT's graduate program in science writing, and his newest book is called So Very Small: How Humans Discovered the Microcosmos, Defeated Germs, and May Still Lose the War Against Infectious Diseases. I'm Ben Comer, and you've just listened to The Business of Biotech. Find us and subscribe anywhere you listen to podcasts, and be sure to check out new weekly videocasts of these conversations every Monday under the Business of Biotech tab at lifescienceleader.com. We'll see you next week, and thank you for listening. Avantor is a leading life science tools company and global provider of mission-critical products and services to the life sciences and advanced technology industries. Avantor works side by side with customers at every step of the scientific journey to enable breakthroughs in medicine, healthcare, and technology. Avantor's portfolio is used in virtually every stage of the most important research, development, and production activities at more than 300,000 customer locations in 180 countries. For more information, visit avantorsciences.com or find them on LinkedIn, X, formerly Twitter, and Facebook.